We need a generation of scientists that is different from the generation we have now, because so it would almost be like if the cement of a building not only held the building together, but also told it how to build itself. So if you wanted to, find a science, someone who wanted to learn about the world, in my opinion, one of the first things you should teach them is...
Welcome to the 15th episode of Apple Finch Pudding, your gateway into the world of science. Today's scientist is Daniel Fried, an upper school science teacher at the Pingry school and the founder of Biochemistry Literacy for kids. Welcome, Dan.
Hi, so nice to be here. Thank you.
My first question is always the same. Do you have a nice science anecdote or a fun fact for our listeners?
Yeah, I have a kind of combination of both. So I'm working in this sort of new space where we're bringing college level science down to the elementary level. So I'm running a lot of these Zoom classes to, help kids and to show that this is possible. And something that came up just the other day, actually just a few days ago, is the very first vertebrate odor receptor complex was crystallized, and the first publication of this came out. There had been other G protein coupled receptors and transmembrane receptors that have been crystallized, but this was the first one about vertebrate odor perception. So the kids in my program, they start off in one of the early lessons learning about odor molecules and what makes things taste the way they do and smell the way they do. So when I made that lesson a few years ago, I said, we don't really know how odors are, actually connected to their receptors yet. It's still unknown. There's this, still a lot to be learned in science. But 3 days ago, this structure was published, and I was so excited to share with these kids who remember 3 years ago not, thinking that we would have this information, and now we do. So it was a really exciting lesson to go over that paper, just like a day after it was published. So I guess the anecdote is, we can be sharing, cutting edge science with very little kids. And, instead of maybe calling them little kids, call them, early science learners or, pre-scientists or future scientists, because, what they are able to do is just as valid and legitimate as what adults can do. So what the takeaway message from that class was, is that, keep watching, and science will develop, and then you will be there to witness it if you know how to interpret it. So it was a really cool, exciting class.
Yeah, that sounds really cool. But also it sounds difficult. Like how do you explain that concept to children?
Definitely not something that you could do to someone who doesn't have any background. So my job has been, how do you create this background, foundational knowledge, so that things like, when a new crystal structure is published, that the kids understand it and that they're also excited about it, because just understanding isn't enough. They have to be very motivated to want to do that. So we can talk a little bit more about the nitty gritty of how do I create these lessons that resonate so well with kids. But, a lot of these kids that were in that lesson have been with me for 3 years. So something about what I'm doing resonates with them, and they want to stay with it. And when we get to these cool endpoints where they can, learn something like that, it's very meaningful for them.
So you're a protein researcher?
Yeah. So that's my background. So I started off at Yale with, I did a PhD in a chemical biology lab, Alana Shepard's lab. And in that lab, I was doing some protein engineering, peptide design and testing. I was working with fluorescent sensors. So that's kind of my background. In my postdoc, I was doing some enzyme expression with the bacterial cellulosome, which is a complex of enzymes that digest cellulose in mainly bacteria. So that's kind of my background. So actually, the goal of biochemistry literacy for kids was to make people understand protein science. That's kind of what the goal of this was.
And maybe to start with the basics, what are proteins? Because you have a lot of different types of proteins as well. You have proteins to build your muscle, but you have also enzymes and stuff. So how would you define proteins if you have to explain it to children?
Yeah, sure. So by lesson, I think it's lesson 6, the kids learn about, or 6 or 7, they learn about amino acids. So amino acids are the building blocks of proteins. This is a particular amino acid called cysteine. There's certain parts of an amino acid that's always the same. For example, there's this ammonium group, there's this carboxylate group, and then there's something else that you can add on to it called the side chain. And those 3 things together basically make an amino acid. And if you connect these in a specific way in a long chain, it kind of takes on a life of its own, starts to fold up and bond with itself. And it can also start to do chemical reactions if the sequence is just right. So that's the idea of what a protein is. So proteins are just very large molecules made out of this particular building block or monomeric building block. Enzymes do all the important things in life. So enzymes are in charge of releasing energy from our food. They're in charge of making all the pieces of our cells, basically. They're in charge of replicating DNA. So they do basically everything. So if you wanted to, find a science, someone who wanted to learn about the world, in my opinion, one of the first things you should teach them is how proteins are structured and how they work. Because if you're like living on the Earth, we live in a biology that's based on proteins. So part of the reason I started biochemistry literacy for kids is like I thought this was like a gaping hole in people's understanding about the world around them. People were going through their whole life probably never really getting much of what a protein was, and proteins are moving my muscles right now. I mean, everything that we see is in terms of biology is protein. So that was really the goal, is to get protein science into the lower levels of learning in school.
Yeah, because I think when a lot of people hear about proteins, the only thing that they can think of is we need to eat proteins. And especially when we want to train and have more muscles, we need to eat some more protein. That's like the main thing people think of. But that's not what you're working on.
Right. So one thing that I did in the beginning to get more Instagram followers is I would always hashtag protein on all my posts. And most people who were following the hashtag protein didn't care about protein folding or amino acids or something. They were just doing exercising stuff. So I got a lot of probably unwanted traffic because but people don't really equate proteins with the structural building blocks of life. I mean, that's actually a lot more important than just like eating a protein shake, in my opinion. So
yeah, of course.
I think the kids agree because it's a beautiful world. If for people who want to be watching this, I can share a couple of the beautiful structures that the kids learn about too, or that anyone can learn about, because that's it's this magical world of three-dimensional structures that are just so alien to us on our level of life. But this is actually commonplace. Like actually, our world is based on these things, and we are made of these things. So that's a great thing to know too.
And maybe also to get back a little on what you said before. So you said proteins are chains of amino acids, but you also said that they fold, so they have a specific structure. Can you tell us a little more about that?
So if you are interested in, kind of being sucked into this world of protein folding, here's a typical folded protein. Okay, this is the green fluorescent protein that makes jellyfish glow. All right. Now, when you show this to a person, the appropriate response is, wow, that is amazing. Can I learn more about it? And that's really what kids automatically do. Not necessarily grown-ups. Not all grown-ups want to learn about that, but kids definitely do. This is something that, you mean living things are made out of stuff like that? Like, I want to learn what all these pieces are. And that's actually the reason I got into protein science too. I saw a structure like this and wanted to learn. So let's look at the idea of a chain. So you can maybe see here, I'm going to try to isolate this, but see there's like a chain. You can definitely see there's chains here. And those little yellow dashes are what are called hydrogen bonds, which are a type of attraction between certain sorts of atoms and electrons. And it's actually what holds the whole thing together. So if you look around the barrel-shaped structure of this protein, you can see that those dotted lines are all over the place. And that's actually what's holding it together. So the hydrogen bonds are sort of the glue that holds together proteins, but it's also what folds it together. So it would almost be like if the cement of a building not only held the building together, but also told it how to build itself. So that's the really cool thing about proteins, because they're self-assembling. So they're produced from the ribosome, and they more or less automatically fold up into a functional machine. And those little machines are what do everything in life. So, I mean, it sounds like, 100 years ago, this would have been science fiction, but now we're living in this time where, just like I said before, every, day, new exciting structures are being, crystallized, and we can learn about, even people in the general population can learn about what these things are doing. Let me see if I can explore it a little bit more with you guys. I have tons of structure. I'm trying to remember which one I brought up here. There's a specific part of the center of this thing. If I zoom up here, see the special little ring here? There's a little ring that has two nitrogens. Yeah. And I've done some extra coloring here, but the greens are just carbons. But this little area of the molecule is called the fluorophore. And this is actually what makes the protein glow. If it makes it fluoresce. So, there's a certain chemical reaction that actually creates that ring. But that ring is from amino acids too. This whole structure is made of nothing but amino acids. There's a vocabulary of 20 amino acids. It's kind of like the alphabet of proteins. Different combinations and sequences will give us, all the diversity of the living world on the earth. So, but this is why we call it literacy. Learn the 20 amino acids, learn the principles, the grammar that describes how to put them together, and then you have a new language. So that's how these kids do it. It's a language-based approach, at least in my mind. And they do have an extra language now. It's almost like a foreign language. And unfortunately, the only people they can talk to is me or each other right now. So we're trying to, branch out and find ways like how can this approach be used more widely? And what can we do with these kids who are now, primed and there's kind of supercharged to contribute to the scientific world? So how can we connect them with people on the outside, not just me, because they need to, get their feathers and fly away at some point. How can other people, collaborate with the program to, utilize these people, which have never really existed before? We've never had kids grow up as biochemistry natives. It's always something you learn later in high school, or, in college or something just like a couple semesters, and then we kind of take care of it. But these kids are, fluent in this language. So they're going to be a really different kind of researcher in the future. So what can we do with them? That's what I'm hoping to get ask that question to more people.
That is an amazing difference. Like, I think the first time I heard of biochemistry was probably at university. And now you're teaching that to children. Like, how old are these children?
The program was started to go into grades 2, 3, 4 or maybe 2, 3, 4, 5 in a public school. And I did hundreds of classes in public schools like that. It really works. I have a few papers about it. Education researchers kind of want, massive longitudinal studies to prove that what you're doing is legitimate. But I don't have the funding or time for that. But I can tell you from anecdotally that this is definitely working. I wouldn't have done 200 classes if it was a waste of time. But it really does work on a general population. Maybe third grade is the best time for it. But during COVID, and because the program sat on the internet, I was licensing it to some schools. But because it was on the internet, it started to attract homeschoolers, and it particularly started to attract highly gifted homeschoolers. So while I always say that the program is geared towards, especially the early lessons, maybe like third or fourth grade, it's attracted some of the most profoundly gifted, human beings in the world. And we have kids 5 years old finishing the program, actually. So can you imagine being 5 years old, completing a survey course on biochemistry? That's a university level designed by a professor. It's really, really crazy. So like, what do you do with those people now? What do you do with a little girl who's just turned 6, who can talk about, beta decay and, Watson-Crick base pairing and whatever you want, from the curriculum? It's crazy.
It is really insane to hear about how these children are grasping these concepts. Maybe to get a little back to your work. So you showed us a fluorescent protein, and you also have some papers on fluorescence. But what is fluorescence?
So fluorescence is when you have the absorbance of light by a molecule. A lot of molecules can absorb light. Molecules, especially this is not particularly fluorescent, but even DNA base pairs. So this is a part of a DNA structure. You can see there's lots of double bonds in it. So double, single, double, single, double, single. That's kind of a sign that the molecule absorbs visible light. And it's sometimes a sign that it's fluorescent. Okay, so certain molecules, fluorescent dyes, have that kind of general structure. Often it's rings, often it's this conjugated double bond system, the single, double bonds. So what happens to these molecules is they absorb one wavelength of light, or they at least a range of wavelengths. It peaks in a certain wavelength. Then some of the energy gets lost, maybe to vibration or heat, and the energy that comes out is lower than what came in, right? Because some of the energy was wasted. So what you do is you can irradiate a molecule with maybe blue light, and then it glows with a green light, which is a longer wavelength or lower energy kind of light. So there's all different kinds and colors of fluorescence, but that's the idea of fluorescence. And all kinds of things fluoresce, scorpions fluoresce and minerals fluoresce. But we have, in biochemistry, we're interested in fluorescent dyes. and maybe even natural fluorescence like that green fluorescent protein. Certain animals have evolved fluorescent molecules for various reasons. So fluorescence is a really cool subject. And the other, why do we care? Okay, fine, animals glow, that looks cool and everything. But we use fluorescent dyes in medical research to make certain molecules glow or watch a certain process happening. You can use them for some kind of biochemical assay. So fluorescence is a really hot topic. It probably always will be, because we can make things light up that would not normally be, easily discernible. A famous example is the brainbow. If anyone wants to Google what the brainbow is, the idea is put a different color fluorescent protein in every neuron of a mouse's brain, and then you can actually see in color all the circuitry and connections of the brain, because each neuron and each axon is a different color. So that's one of the applications, is seeing things that would otherwise wouldn't have any contrast to them. You couldn't tell what was going on.
How is fluorescence linked to your protein research?
Yeah, so when I was in my PhD, we used a fluorescent dye to monitor protein folding. So that was the original research that we did, my research team. So what we did is we made a series of small peptides, and we used certain we used this amino acid, coincidentally. We used cysteines, which had a certain affinity for another fluorescent molecule. So if you had the cysteines arranged on the protein a certain way, the fluorescent molecule was a pro-fluorescent molecule. It wasn't quite fluorescent yet, but when it bound to the peptide, it would become more rigid, which allows it to become more fluorescent. So basically what the experiment was, proteins that are more folded will glow more when you engineer them this way, and proteins that are less well folded will not glow as well. So it was kind of a proof of concept to say that we can use this fluorescent molecule to monitor protein folding. So theoretically, you could use this technology to learn about to research protein folding diseases. Certain diseases where a protein is mutated and it's not folded correctly. So you could theoretically use that assay to look at real life problems.
Based on fluorescence, you wanted to decide how much a protein is actually folded, and link that to something else like a disease or something.
That's what it could be used for. Yeah. So the main work that I did was on the proof of concept, figuring out how do you actually make a protein glow when it's folded and make it not glow when it's not folded.
And fluorescence is something we find often in nature, actually, like a lot in ocean life. Do you know why that is?
It's still a matter of debate of why fluorescence shows up in the ocean, and also bioluminescence also, right? Could have something to do with, we know that some famous bioluminescence is used to lure fish to another fish for food. But why do things fluoresce, though? Could be just coincidental. So there could just be molecules that are produced maybe for sunscreen or protection against UV damage, just happens to fluoresce. But when a molecule fluoresces, it's kind of wasting some energy, like I said before. So energy comes in, some is wasted, and then it gets emitted at another wavelength. So that might be the reason that we see fluorescence in fish and other things, because if you're, sitting out in the sunlight, in kind of shallow waters, you're getting irradiated all day long. Maybe you need some fluorescent molecules in your skin just to, act as a sunscreen in a way. So that might be one reason, or there could be reasons for communication. Certain fish may have eyes that are tuned to see the fluorescence of, the patterns on other fish and things like that they know of. So they still don't really know, because we can't interview a fish, because then they could just, hey, why does your friend fluoresce? Tell us about it. But so those are some of the reasons that fluorescence shows up in the ocean. One interesting thing is, as you go down, obviously, into the water column, most of the wavelengths get filtered out. You're left with basically blue light. So if you've ever gone snorkeling or scuba diving, everything's more or less blue down there. It doesn't look very impressive. But you can imagine that if a fish was fluorescing green and it was producing its own green light at a depth, that might be seen and might stand out among the other fish at those depths. So that might also be a reason. We see a lot of fluorescence in green or blue, or sorry, green or red. So those colors would be very uncommon, several feet down or several meters down into the ocean. So that might be why we have fluorescence also.
So you mentioned fluorescence and also bioluminescence. You also have phosphorescence. What is the difference between all those terms?
Fluorescence is a sort of a passive process. So a molecule will become irradiated by sunlight and then waste some energy and then emit a lower wavelength, different color. So that's a very passive process. The bioluminescence is the process where you actually are using energy to create your own light. So you'll actually the cells will be consuming ATP, or they will have some kind of molecule that's getting used up in this process. So you actually have to be constantly creating new molecules that feed the bioluminescent system. So that's kind of like thing you see with like a firefly or a deep sea angler that is actually producing its own light. Now the interesting thing is that you also often see a combination of fluorescence and bioluminescence together. For example, the jellyfish that produce that green fluorescent protein I showed you before, that GFP doesn't operate by itself. There's also a bioluminescent molecule that works in concert with it to produce light. It produces blue light, but then that blue light gets shifted to green because of the fluorescent molecule. So there can be a partnership between bioluminescence and biofluorescence to not only to produce blue light and then turn that blue light into green light. So that's what the jellyfish do. So there's some reason that the jellyfish is producing the light because it's very intentional. It's not, oh, I just happen to have a fluorescent, like the scorpions. We don't really, I don't know. Do scorpions need to be fluorescent? But the GFP, it's a very targeted, process there.
Yeah, that's really insane because you can think it transforms the natural light, but it also produces its own light to produce or to reflect. So that's really interesting. It's like in the absence of natural light, I'll just produce it myself. That's fine.
Yeah, that's what it is. Yeah.
And also in 2021, I think there was this whole thing about all the animals they found in musea actually that were fluorescing.
Oh, in New Zealand? In where?
I think it was in New Zealand. I'm not completely sure.
Oh, I didn't know this.
Because it was also the platypus and stuff like that. They were all stuff.
Oh, yeah, right.
Yeah. That was a whole thing because they started searching the museum and a lot of animals that they didn't expect were all fluorescing and they don't really know why.
Yeah, it was definitely something that happened in the last 10 years where people started to realize, and I know a couple of the people who were doing this with marine organisms, but yeah, things that people didn't know were fluorescent, they just started to shine a light on them and then they realized. So yeah, theoretically you could go out and explore, a jungle somewhere and find some new fluorescent stuff. Could happen.
There's also been a lot of genetic manipulation to make animals fluorescent. Is there a practical reason for that, or is it just something fancy that people want to do?
Yeah, so if you ever want to see the genetically modified GFP, just go to any pet store. You'll see the GlowFish, which are the fluorescent fish. They're not naturally fluorescent. At least here in the United States, they're in every pet store. And you buy fluorescent lights for the fish tank, and there's usually fluorescent rocks too. But the fish are fluorescent, and they've been genetically engineered. So if you remember a minute ago, I showed you that fluorophore, the central piece of the GFP. You can actually modify different amino acids nearby. For example, this ring here, this is a tyrosine. If you change that to other things, you'll get all different colors. You'll get cyan and you'll get yellow. Yellow is a sort of different process. You can get blue ones. So you can get blue FP, cyan fluorescent protein, all the other, I said blue FP, BFP. So that's how that works. The mutant changing a fluorophore to change the color. Practical reasons, let's say you wanted to look at a cell, but you wanted to see how one protein and another protein interact. You probably need to make one protein one color and another protein the other color. And then when the two colors come together, you know that their protein's interacting, for example. Or you can tell, how about this organelle is fusing with this organelle or something like that. You could theoretically look at those kinds of changes if you have multiple fluorophores. There's all kinds of other reasons to have different color fluorophores, but those are some of the easy ones.
Do you still work on fluorescence or do you do something totally else now?
Yeah, so I was a professor for a while. I got tenure, but then during the pandemic, I decided to change what I'm doing, and I'm actually working in a high school now. We have a really beautiful research lab, though. So they actually do basically college-level research at this high school. So I'm able to still do protein expression work. So I'm working on some fluorescent protein expression of some fish that I have collaborators who have basically found these fish that are fluorescent, and they know the gene that causes it to be fluorescent. So I'm working to express and do experiments on those proteins. So actually doing the same, almost the same kind of level of work that I'd be doing in the college also.
What is the current goal of your research?
So these proteins have never been expressed. These are vertebrate fluorescent proteins, which are all very new. Like I said, like you mentioned, finding fluorescence in vertebrates is kind of a new thing. Like you said, going through the museum and finding everything. So it's never been expressed. So trying to express it theoretically, we could crystallize it, see how similar it is to other things that have been expressed. There's another eel protein that's also been crystallized. So there's this new world of vertebrate fluorescent proteins and other interesting proteins that might be worthy of exploration.
What are some of the major challenges currently in your work?
Well, so like I said, I'm mostly doing my teaching now. So this is, I'm not sitting in lab for 18 hours a day like I used to. But the challenges are, typical protein expression challenges. Solubility. These things don't necessarily want to be expressed in bacteria. But it's going okay though. We have protein, so we'll keep going with it. It's really interesting working with high school kids training them because it's the first time they've ever expressed the protein. So it's like an eye-opening thing for them.
Maybe we can go a little bit further on that teaching. Like, where does that drive come from?
Yeah, so I like teaching anyway. I'll teach anyone about anything. I like teach drawing or music, whatever. I just like helping kids and understanding how their minds work is really interesting to me. But the drive to dedicate 10 years, basically all my time since graduate school, all my free time, to giving little kids a window into the world that I only learned about as a graduate student. There's all different motivations for it. We can get into what each one might be, but I guess the most outwardly interesting one is that we need a generation of scientists that is different from the generation we have now, because the progress is we're progressing so quickly. It's like an exponential growth of scientific knowledge. But in school, it's almost unchanged what the kids learn. So the divide between what the kids come out of high school or college knowing and what they have to do as someone to contribute to science in the future, it's just getting larger and larger. What's going to happen in 100 years when we're still learning what H2 atoms are, and what CO2 is. And then we have, nano architecture robots making things for us. Like how do that something has to change. We have to really, really change what's going on. And it has to be faster than, the pace of education research right now. It can't be these like little steps. It has to be like a big step. You have to take a big leap of faith that other things can be, that we can make a big change, and it just has to happen right away. So we can analyze how it's going later, but we have to do something quickly. So that's one big motivation is just, I felt like I was very unprepared for graduate school. I was lucky enough to get into graduate school, a really good graduate school, and I got into a really good lab. But I just knew, like, once I started, and you could tell from other people too, that like we should have really done better in the lower levels, because there's so much time. You have 12 years. That's an enormous amount of time to get stuff done. And I didn't know basic stuff that I should have known, that I felt I should have known to make a contribution. So like I'm filling in all those gaps myself. I wish that I could get other people to realize that this is something to do. But at this point, I'm doing it. I'm sure there's other people doing it. I just don't really know where they are. But that's a really important part. We need like a rapidly improved K-12 system where kids are, and also that goes to teachers too. Like it's also teacher training, because the teachers are not well trained either. They may take one chemistry course and then become a chemistry teacher or a science teacher in the elementary level. So this is also a way to make teachers more comfortable, because I wouldn't be comfortable teaching chemistry if I took, intro Gen Chem course in college. I wouldn't be able to teach chemistry. So this is a way for making teachers feel more comfortable also, if they see how fun it can be, and how, it's connected to everything. So that's one big thing. So just trying to make a change to the world, that's a big one. I just really like doing it. I like learning stuff that I never knew before. So this is just a way for me to, like, I'm doing a unit right now on food coloring and pigments in nature. And this is not something that, you really study very much, but it changes the way you look at the world. Like every food, I know that the pigment that makes that drink blue now. So that's going to be a really fun unit for the kids to learn too. So I just like learning stuff, and I like answering these questions that are not necessarily like MCAT questions or Chem GRE questions, just questions about life and like how things are. And that's what I feel like is also missing in chemistry curriculum, is just like the playful nature of, oh, I wonder why that is purple. I'm going to try to figure that out. And there's no, analysis of it. There's no, math problem that I have to do for it. It's just trying to take in what you see around you and learn how everything works.
I really recognize that drive, because actually my main drive for doing this podcast is because I love learning new things. I love science in particular, and I want to share what I learned. So this way I'm actually learning and sharing at the same time.
Totally. Yeah, same for me. Yeah, that's why I'll be, one day I was reading about hemoglobin, and then I stumbled across the worm hemoglobins, which maybe that might be something for if some of your listeners want to look into worm hemoglobin, which obviously carries oxygen in the blood of the worm, but the worm hemoglobin is so much bigger. It's amazing. So our hemoglobin has 4 protein components. The worm hemoglobin, I forget how many, has like 100, a couple hundred or something. It's gigantic. So it's like a gigantic hemoglobin that binds so many hemes and so many oxygens. So I learned that, and I was like, I got to tell the kids about this. So I made a quick lesson about it, and then that day, I told, and that's like their favorite lesson. Like they still talk about worm hemoglobin, particularly the giant tube worms that live in the deep sea vents. They have a giant version of hemoglobin. So so just sharing what you know and creating an opportunity to learn. And I've learned so much by doing these classes and by creating this curriculum. One thing that I always wanted to know, ever since I was in high school, I remember I was interested in how come there's a certain isotopic distribution of the elements. For example, there's a lot of oxygen 16, which has 8 protons and 8 neutrons, but only a little bit of oxygen 17. And oxygen 17 is stable. It's not radioactive. So why is there so little oxygen 17, but so much oxygen 16? So that's true for every element. So there's got to be something there. Like, why is there one and not the other? So I made this giant lesson on how stars create the elements, stellar nucleogenesis. So this is also one of the, crowd pleaser lessons that the kids love. It took me a couple of years to make this lesson, just this one lesson. I had to do all the research, but we answer that question. Why do stars make only oxygen 16 and not the others? Why is so much carbon 12 made and not carbon 13? So it's just amazing to get to that, to go spend your whole life learning chemistry, and then later in life, like, see, okay, now I understand the isotope distributions, and it's connected to how they're made in stars. Like everything we do here on the Earth is somehow connected to this vast history, and it's that profound, understanding is not lost on the kids too. They totally get, how connected that makes them feel to the universe.
I'm sorry, but you have really piqued my interest. Can you explain the link between stars and the isotopes?
Oh, yeah, sure. Let's say I wanted to do carbon 12. Okay. So on the left, we have 3 helium atoms. If you have 3 heliums in a star, and this happens in stars all the time, there's heliums there. If you take those 3 heliums and fuse them together, it makes a bigger atom. Okay. And this is what happens. This is the job of stars. What stars do is, because of their immense gravity, they crush atoms together and they can form larger atoms out of those things. So, I mean, this is kind of a very obvious one, but look at our heliums. We have helium. Each helium has 2 protons in the middle. So the big red ones are protons. So there's 2 protons and 2 neutrons. Neutrons are the gray ones. So 2 and 2. So if I add 2 protons plus 2 protons plus 2 protons and 2 neutrons and 2 neutrons and 2 neutrons, I get 6 neutrons and 6 protons, which is a carbon 12 atom. So that's why there's so much carbon 12 around in the universe and very little carbon 13. So that's just the very beginning one. A lot of them are much more interesting. You're also noticing something really interesting on the bottom here. We have the masses. Okay, so if you look on the periodic table, you see all the masses. This also confused me like endlessly. Why is the mass of a helium 4.002603, but the mass of a carbon is 12? Carbon 12 is, why is it a round number? These are kind of answers that we can get into. But something really cool that happens, watch this. If I take three heliums that have a mass of a little bit more than 4, and I fuse them together, I don't get something that has a mass of more than 12. it's exactly 12. Okay, so carbon is exactly 12, but the things that make up a carbon are more than 12, right? It's 4.02 times 3. So what's going on there? It should be 12.078, but it's not. It's just 12. So what is that? That's the missing mass that we are releasing when we do fusion reactions. So some of the mass that the heliums comprise of is being lost to energy, and that's what starlight is. So stars are doing two things, and nobody knows this because it's not. No one talks about it. It's not in the curriculum, so it's not talked about very in much detail. But stars are doing two things. They're taking small atoms and making them into bigger atoms. So they're creating heavier elements, which we need to have life and things like that. And it's also releasing energy in the form of light, which is what starlight is. So we're making both the stuff of the universe and the energy that can power life also. So I just I just feel like everyone should know star. I mean, I'm not a astrophysicist, but I just feel like everybody should know some star chemistry because it's so cool and just connects people. It connects you so much to everything else in a way that's even more than biology in a way.
I can imagine that children are baffled by it, that they must love this stuff.
They love this. Yeah, they don't actually. There was one kid that was like, "Are we done?" Like, was that 5 weeks later? I was like, "Are we done with star chemistry? Like, we want more star chemistry?" So, like, no, sorry, we have to go back to organic chemistry now.
Yeah. and like in terms of organic chemistry, what do you teach them then?
I've done a lot of experimenting with organic chemistry. Back in the very first lessons, I thought that I could teach kids electron pushing right away because I liked it so much. Using arrows to show how electrons move around. If you don't know, if you're an organic chemist, what you're good at is arrow pushing, which means you take this is a good model for it, but you take an electron pair and you show how it moves using an arrow, and that's how you make a new bond or break a bond or do an acid-base reaction or something like that. So that's what organic chemists do. I tried doing that in the very early classes. It didn't work. It was one of the few things that I couldn't get kids to do. I could get kids to do almost anything else, but electron pushing was hard. What I did is, after teaching hundreds and hundreds of classes, I've come back to it. And I'm still working on how to actually get organic. I mean, the kids are learning organic structures for sure. I mean, they understand bonding rules, but actual organic chemistry where we see how reactions work, that's a harder level. But my advanced kids can do that because they really, really internalize what a bond is. Once the kids really get what a bond is, that it's an electron pair, and they get some concepts of like electronegativity and some acid-base chemistry. Once they know what molecules are going to do, then they're ready to understand how they do it. So yeah, I use an organic chemistry textbook and we do whatever reactions that are interesting to the kids. So yeah, the kids can do organic chemistry too, no problem. But it's a little bit more of an advanced level for them. They have to kind of be very familiar with structures before we get into actual reactions and things like that.
Yeah, but this project of yours has become immense, right? It's not only in the US, it's worldwide.
Yeah, I mean, I wish it was immense, but we have about, I don't know, it's probably around 2,000 homeschoolers.
That's pretty big.
Yeah, and that's, that's not like a YouTube view. That's like someone who like spent like $170 to like access it and buy the model kits and ship it to themselves. So and they do it. So yeah, it's really an honor that people would like trust that I can help their kids in that way. It's really like every time I get an order, I'm like, it never really goes away, that like feeling that you're doing something helpful for someone and you don't really even know what that person's going to do with it. I mean, even a weak student, like I wasn't a very strong student actually, even a weak student when they're 8, they could do something completely crazy and profound when they're 25. So it's not even about like, oh, this kid's a genius right now. Look what a great genius they are. It's like, oh, this kid did 3 lessons, but they weren't so great. They kind of took a pause for it. But that makes an impression still on them, and that could do something else later in their life. But in general, though, these kids do a lot of work with the material. They get very far, and especially the kids that do the Zoom classes with me, they're with me for years now, and I can see them develop. I can see how they, I really know what they know, and I always enjoy bringing other educators or scientists to see it in action because it's really like takes your breath away, like what these kids can do. And how it really could be everywhere where this is happening, but we just need more care or more buy-in from administrators or people in power. But yeah, it's really cool. We have kids in Australia using this, a couple hundred in Australia, and then thousands all over the United States and Canada. And like one or two in every country, and not every country, but maybe like 45 or 50 countries. So it's really cool, sending the stuff to all over the world. Never thought that I'd be able to do that.
Yeah, and I believe that is having a great impact on those children. And maybe also just what is your take on, like you said yourself, you were not the strongest student when you were younger. Often people or parents actually or teachers go like, oh, this is not a strong student. We should not give them the hard problems. But I don't think that's always the best decision. But what is your take?
I agree. So I had reading issues. I had just decoding issues. So I was felt like I was smart, but I couldn't finish reading anything. I was always the last kid to hand in anything. I was always a very good writer, but I couldn't really read a book. It was like really a trouble for me to read a book until like college almost. So really put me at a handicap all throughout K-12. And I'm sure it changed what I was able to do with myself because I couldn't handle the amount of reading. I remember on the SAT, whenever there was a paragraph to read, I wouldn't even do that question because I knew that it would take me 10 minutes to read it. I have no hope of answering it. So I was already, taking something like the SAT, I already couldn't answer. I don't know how many questions that would be, 20 questions. I already just couldn't do at all. So, it's weird because the people approach me, they're like, oh, is this curriculum for geniuses or something? I'm like, no, I'm not a genius. I couldn't even read when I was 8. So I'm designing this for someone who can read like a few words at a time and uses a picture because that's how I got around everything. I used pictures and I did things that were more visual. So it's written for someone who almost like has a learning disability in a way. It's not written for geniuses, but if you are profoundly gifted, the visualizations just become part of you. I mean, the kids don't forget what these things are. So it works well for all learners, including someone who might be a little bit slower or has, comprehension issues and things like that. So that's who I'm trying to reach. I'm trying to reach everybody with it. It's definitely not some kind of elite program. And the whole point of it was actually to make these subjects more to kind of democratize it and to make it so that we didn't just have some kind of elite kind of kid, climbing way to the top, leaving everyone else behind. This is really for everybody. But unfortunately, you need schools to kind of collaborate with you to get access to those kids. So that's one of the challenges that the program has had is, how do you, I can make the thing better and better and better. But at some point, you need grown-ups to also collaborate with you. I can work directly with the kids, but the grown-ups have to help. And the homeschool parents, it's an easy sell for them because they see, oh, I want to help my kid. Let's just do it. But for schools, it can be, there can be some bureaucracy. So finding out how to overcome that is something that has to be done in the future.
They need to see the value of your project, actually, which is hard when you're still stuck in the old system of teaching.
Yeah, but some schools have done it, and they've done it successfully for multiple years. So it really does work. It's just, there's limited time that the schools have, but it really does. It's going to take something from the top to say we need to try some really, really drastically new stuff. Otherwise, it's just going to. I mean, it's fine if they want to keep it the same way it's always been. We'll just have, it'll be certain populations that are doing programs like this, and that's just the way it will be. But one way that I'm going to try to get the word out about this is by doing this summer camp, which I'm not sure if I told you about it too much before. I think I did tell you a little bit about it before. But we're, one of the ways to get people to see what this is, because it sounds probably pretty crazy the way I'm talking sometimes. We've documented a lot of the programs. So I've had 2 little documentaries made about one was in a public school, one was in a private school. They're on the website. And then what I'm going to try to do this summer is to create more of like a feature-length documentary that follows certain of my really great students that have been working with me through the Zooms, follow them to the summer camp that I'm running here in New Jersey, where all these kids who have been sort of trapped in Zoom world for 3 years, we're all going to come together. And, kids are flying in from Australia for this, and they're flying in from California to meet each other and to be together and to do some practical biochemistry research and physics research, activities, lab activities, and just to kind of network and meet each other, because it's it's going to be really, really cool to see that. So we want to document it. So we'll have a documentary hopefully made that shows, just what this means to the kids and how it's really part of their lives now and part of their future, because they all want to help the world with this knowledge somehow, because they know this is a special way that only they can, that only they have to help the world in a way. And
can you tell us a little about what you're going to show those children that are coming to your summer camp?
Yeah. So in the camp, they have a very strong theoretical knowledge. We've been doing organic chemistry and biochemistry. They are fluent in protein structures. They can use PyMol. If any people are in science research, they probably know what PyMol is. It's that 3D viewing program I just showed you before. They can do all those things, but they've never actually held a pipette. They've never made a solution of something. They've never weighed something on a balance. So it's just really crazy that, they are, it's almost like, I don't know what it's like. It's almost like a Benjamin Button or something, where they're like, they have an old mind, like a sage-like understanding of chemistry, but they've never actually weighed something before on a balance. So this will be their chance to do some practical stuff and to feel like they, they can express a protein, they can do a DNA gel. So we'll just kind of do some fun, we'll transform bacteria to make a fluorescent bacteria with the GFP, like I showed you before. So they'll do some, common lab manipulations. And then I have another collaborator also a former professor and physics teacher. And she's going to be doing quantum physics experiments with them, like double slit and things like that, because the kids who love chemistry also love quantum mechanics. And it's really weird. They all want to know quantum. They all want to know about quarks and stuff. And I'm like, I don't know about quarks. Like, you have to find someone else to tell you about quarks. But yeah, the kids who get into this, they want to get even smaller. So we're going to give them some quantum also. We're also doing a day where we go to Princeton University for them to do poster presentations. So they're doing some kind of like literature review, and they're going to make they're going to have little groups that will make posters, and then they're going to present to the faculty at Princeton, which will be really cool. It'll be their first chance talking to other scientists, really, besides me. And hopefully, there's different things that could happen from that, but it makes them feel seen, because a lot of these kids who are doing the program, they often are not believed, sometimes by their schools or sometimes by, other adults around, people they're trying to collaborate with. They don't even believe that they really can do these things. So, the issues that I had in the beginning, where people didn't believe that I could teach these things, now the kids have the same. I've unfortunately passed along my problems to them. Now people are having trouble, taking them seriously, because it's just so wacky, really. when you say, I study protein folding, blah blah. And I'm 7 years old. So we need visibility, and we need for them to be seen and taken seriously, and something has to be done with them, because someone who can do all these things and is not allowed to is a waste for the society, but also, psychologically, like they need to be doing something, and they need to be feel like they have a meaningful existence, and that what they know can be, used by something.
Yeah, and if they can do that stuff at 7, what they'll be able to do when they're 20, I don't know. It's
hard for me to know either. Do we want everyone to be a scientist? No, not everyone has to be a scientist. But what if everyone had a literacy and a sense of wonder and an appreciation for biology at a molecular level? What if when you went outside, you didn't just say, "Oh, that's a pretty leaf"? What if there was something more there? You know what I mean? When you listen to music, if you're a musician, you have a deeper understanding of the theory that goes into it, the appreciation of how hard it is to make that instrument, how to play that instrument. So it's the same thing. Like, why can't science appreciation be something that we want to give everybody? Not everyone has to be or can be a researcher at Harvard. So there's other levels of this too. Can policymakers make better decisions if they knew what molecules were? There's all kinds of reasons to educate the public about this kind of stuff.
I totally agree. Not everyone needs to be a scientist, but you need to know the possibilities. And then you can make a decision for yourself, what you like or what you don't like, and what you're good at and stuff like that. If I heard you correctly, so yeah, it's biochemistry literacy for kids, but you also talk about quantum physics or someone else in the team. So it's going in other branches as well?
Yes. So my program is, it's an introduction to chemistry, biochemistry, and organic chemistry. And then I also have some other things like nuclear chemistry is part of it also. So I'm really more of I kind of stay in the things that I have some training in. I don't think I will ever have the background to teach quantum physics. So there's another person, Azadeh Samadani, who's also at my school. She was a Brandeis professor before she was a teacher with me. She is really a pro at teaching kids too. She appreciates the need for bringing this down to the lower levels in school. So she's been kind of my collaborator. And at the camp, it's going to be great because the kids will have, they'll have like a morning of biochemistry, then like the afternoon will be, quantum physics basically. And that would be more, they're not all super trained in physics, but, it's just kind of like a fun for them to get a toehold in that world.
I'm sure those kids will have a blast.
Yeah, it's going to be amazing. There's all different, dimensions of what it could be. But, imagine kids who have collaborated with each other on Zoom, who have learned together and can speak a language that basically no one else around them can speak, only their friends on Zoom. And now this is the first time that they're able to speak to each other in that language in person. It's just going to be like crazy to see, what happens to, these kids from all over the world who've known each other and now are find their family and their home. It's going to be really cool to see. So I'm really looking forward to it.
I truly believe that. We're going to round up around here, but is there like a take-home message you want to share with our listeners?
Yeah, let's see. So the take-home message is don't be satisfied with the status quo. Look to see what's possible. You can challenge what's happening. And even if it's a little uncomfortable, or even if people don't believe you at first, if you know what you're doing, you can prove them wrong and you can make a positive change in the world. So, the establishment is there to stop innovation, unfortunately. So everyone else can be here to do the innovation. And that's my take home, I guess, is to just keep trying, even if it's frustrating. I know that's a cliche. People told me the same thing when I was trying to give up. But if you have something good and you don't give up, eventually it starts to, help people.
That's wonderful. Okay, this was the 15th episode of Apple Finch Pudding. I want to thank Daniel Fried for the information, and let's meet again for the next episode of Apple Finch Pudding.